The topic for my today's talk is about tau422 and our neuropathological biomarker for detecting mild cognitive impairment of Alzheimer's disease. It's well known that Alzheimer's disease is insidious onset neurodegenerative disorder with gradually. Sorry, thank you. Yeah. All right. Gradual decline in memory and learning. So the pathological changes, including the intracellular neurofibril tangles formed by the hyperphosphated tau and extracellular A-beta amyloid plaques. So MCI represents the early stage of AD. For people older than 60 years old, around 12 to 18 of them are living with the MCI due to AD. So within 5 years, 30% of them will develop AD dementia. So within 10 years, this percentage will increase to 50%. So if we can detect the AD early stage, we can provide early medical intervention. So you might prevent or slow down the cognitive decline. For example, so some cholinesterase inhibitors have some effects on slowing down the disease progression. So unfortunately, so far, there's no cure for the AD dementia. Therefore, for finding the new biomarkers for MCI could help to provide more effective prevention and treatment for AD. Researchers are looking for the biomarkers in CSF or plasma or through the genetic screening or the PET imaging. For some of the familiar or sporadic AD, we can screen the mutations in gene amyloid precursor protein, PRIS91, PRIS92 genes, or check the ApoE allele carrier to predict the AD risk. Changes in the A-beta-42, A-beta-42, ratio, total tau, and phospho-tau in CSF occur before the AD symptoms. So theoretically, these markers should be good for detecting the early AD. However, CSF collection like the PET imaging, the invasive and relatively expensive. So this limited the clinical application. Recent research found several phospho-tau biomarkers in the blood samples. They're like the phospho-tau 181, 217, and 231. So the rationale for this study, we already know the neurofibril tangles, they're formed by hyperphosphated tau. Then these neurofibril tangles deposit in the hippocampus and the entorhinal cortex, then the spread to other brain regions, then finally reach the temporal cortex, parietal cortex, then cause the cerebral atrophy, and thus induce the cognitive decline. So based on the site-specific phospho-tau frequencies in AD, we performed site-specific phospho-tau antibody screening to validate those sites in the brain tissue. So we identified several site-specific phospho-tau sites, p-tau422. So p-tau422 is one of the candidates. Here's our Western blot results. We use 3 known biomarkers as a control: 217, 231, and 181. So the high molecular weight smear stands for the tau aggregates. In AD group, we can see here in AD group, the high molecular weight smear in AD group has higher intensity than those in the control group. Especially the p-tau422 has higher specificity than other markers we can see here almost no signals was detected in the high molecular weight area in the for the control group. So to further study the phospho p-tau422 in MC brain, we performed the ELISA assay. Here's our results. So from the scatter plot, we can see here the p-tau422 levels in MCI brain increased almost 11-fold when compared to the with control. So others only increased, other markers only increased 1.5 to 2-fold. So we also made the ROC curve to analyze the specificity and sensitivity for those markers. So our p-tau422 has higher sensitivity and specificity with the AUC area on the curve, 0.91. So compared with the 217, it has only the 8.3, others even lower. By collaborating with Dr. Jerry Wang in Duke, we further validate the phospho-tau422 expression in MCI brain using immunostaining. Here's the results. So left columns show the cognitive normal brains, right panel stands for the MCI brain. So our results show that p-tau422 has higher denser tau pathology in the hippocampus and the surrounding brain regions. We can see the in the MCI brain, for the hippocampus C1, C2 regions and the subicular regions, you can see obviously they have higher density in MCI brain compared to the control. So we quantify this signal and made the whisker box plot. Our results show the phospho-2 has higher density in the MCI brain compared to the control in all the test brain regions. So summary, our result demonstrated the p-tau422 differentiated the subject with MCI from cognitive normal control in brain. Brain tissues p-tau422 immunostaining results confirmed our findings. For my rec proposal, so we just validate that our biomarkers with larger sample size, or then extend to CSF and plasma testing. Signal-wise, to develop more MCI markers. So we already have some candidates already. So next step is to validate the larger clinical cohort and then develop the ultra-sensitive home blue CMOR tests to detect the phospho-tau and other candidates in the CSF and the plasma. So finally, I would like to thank all the lab members in Dr. Xu's lab and our collaborators, Jerry Wang and Andy Liu in Duke, Dr. Zhu in Case Western, and George Bloom in UVA. Finally, thank you for your attention. And questions for me?
There's so many sites that are phosphorylated on tau, and I wonder what's so special about this 422 site compared to, 30 or 40 other sites.
This site in the C terminal, so it's the I can actually compare the N terminal one, in blood samples there were less rich abundance, but people have the they check the 396, also the C terminal, some of them detected. So for 42, for our results, so we tested with the brain tissue. So this one is extremely good performance. We get the AUC in AD for 1. This would never happen in our experiment. Others, 0.9 something. This one is 1. So.
And when you did the follow-up staining in MCI and different Brock stage cases, do you see this increase in S422? by histology?
Yeah, from the immune staining, yeah.
Yeah.
From the early stage, the p-tau4 staining, yeah, MCI brain. Yeah.
And it continues to go up as disease progresses, is that right?
That's why I think it's for the brain tissue, we cannot do the disease progression. We just, one time.
Okay,
Yeah,
thank you.
From the brain. Yeah, thank you.
Thank you so much, Dr. Wu. I wanted to ask, depending on, I guess, the amount of the 422 phosphorylation tau aggregates, do you expect to see differences in cellular morphology? For instance, like with different, I guess, phosphorylated tau?
Mm-hmm. We haven't reached that step yet. Yeah, thank you. That's a good point. Maybe we can, pay attention for later.